fellow will then enter the nucleus and change whatever it has to and cause the protein. And the method, of course, is to transmit it intravitreal, but also subretinal. Um, it's not always working. This is being worked on. So the gene delivery does involve placing a strand of DNA, as I mentioned, in a virus that normally enters the human cell and can drop off the gene. We talked about herpes simplex virus. And there's a condition that it's being treated, and that is not the retina, the cornea. And this is being done at Bascom Palmer. It's formulated into an eye drop version, remove the scar of the cornea, and by putting this repair in it, this is done by Dr. Alfonso Sabeda. Um, this solution can replace the genes and clearing of the cornea. It's unbelievable. It's still being worked on. I feel that the coronavirus could be helpful. Watch this. You put the replacement gene in the spike in that, and that could transmit, because we know how coronavirus could affect the human body, everyone's body. But let's put it to use. It could break through the membranes with the repair gene and hopefully reverse it. Then we have non-viral strategies. Just give me a minute. The ribosomes, the antisense, oligonucleotides, etc. I want to mention CRISPR because Jennifer Doudna won the Nobel Prize for this. Here's what CRISPR stands for, and what this does with the Cas9, it could cut out segments of the gene of the DNA, repair it, and hopefully get what we want. It's used with the Cas9, that's the enzyme. Of course, this can be controversial if doing it to human embryos and therefore making people that we want. Here's other methods: electroporation, high-intensity electro fields, and iontophoresis, low voltage that could change the current. Space flight, yes, it's in the papers. Ionizing space radiation and cellular DNA alterations, not only in the whole body, but also in the cornea. And our space people who were up there for what, almost a year? Believe me, they're being studied for this because it's been shown in rats that were in space for quite a while. Now, I mentioned the elephant in the room, and this is one of my colleagues at Cornell that does work on AI and genetics. And the base programs models really can revolutionize how we think about biology and how we develop it. And there's a lot of AI papers that show about the DNA sequences. But in the Eye journal a few months ago, there's a warning about it, the modern threats in academia. And I discussed this with my professors at Cornell about the papers that are being put out by the students and fellows and how much was done with AI and how much from the brain. So anyway, here's my references. And we're off to the future. Thank you.
Thank you, Jerry. Anybody has any questions or comments?
Thank you, Dr. Schultz, for that excellent talk. Thank you for making it so simplified. But my always, as a clinician, query is that, most of these gene therapies are like targeted therapies for specific genes. Like CRISPR is still a little bit more, like you can use it more widely. But which other therapies do you think would have a more wider application? Like we don't have to depend upon a particular genotype or a particular gene and specifically target those kind of diseases.
Well, this has been bothering me too, because how do we get into the gene that's offending? We know what could be done, but how can we do it and be specific? Now, with the retina, we could put it under the retina and maybe that will change, and hopefully the replacement genes will multiply and knock out the offending genes. Yes, you hit right on the problem.
Kumar, yeah.
The answer to my question is that there is currently a lot of traction on agnostic therapies or non-gene dependent therapies, which are anti-inflammatory in nature or anti-apoptotic in nature. So these are not specific for a gene, and therefore it can be easily scaled into a good business model, and therefore can be applied universally. So a patient with what kind of mutation doesn't matter. So they start off with inflammation, then you have oxidative stress, then you have apoptosis. So you try to manage your patient with all these kind of multiple therapies. So that's how the field is moving.
By the way, I want to invite all of you to our session tomorrow morning on ophthalmic genetics. We're going into the basics and expanding it. 8 o'clock in the morning, the basement room A, whatever that name is, the
Amaltas.
tongue twisters, but it's down there, 8 o'clock in the morning.
Amaltas. So I'll invite Dr. Jenny. Dr. Jenny, next you can give us. Yeah, yeah. So next we have Dr. Jenny Weeks to speak from Harvard. So you have to sign, Jenny. Okay.
I see them here, but I don't see them. Oh, great. Okay, thank you.
Yes, sir.
All right, thanks very much for inviting me to speak in this session. You know, I really appreciate the collaborations that I've had so far, starting really with Kumar 20 years ago here in India, and then some collaborations with Ronnie George at Shankar Nathraya, and now Dr. Vinny Gupta and I are starting a new collaboration here in India, which is very exciting. But for today's talk, I'm going to talk about a couple of collaborations that actually have concluded. These are disclosures which are not relevant. So just, you know, we all know that glaucoma is a very important disease, common cause of blindness, and that disease screening and risk stratification are challenging. And as a result, many people have glaucoma and don't know it. But genetics can actually help this problem because genes that cause or contribute to glaucoma can be used for genetic testing to identify people who are at risk for disease, and can also be used to identify novel targets for gene-based therapies, something that we were just hearing about a moment ago. To find these genes, it's really been helpful to form these large-scale consortium. And this is true not only in glaucoma, but really across human genetics. Consortium have been extremely useful for gene discovery. And there's several reasons for that. First of all, the consortium brings together people with the necessary expertise. So these kinds of genetic studies require somebody who's an expert clinician, who can do phenotyping, requires a team to do molecular biology and bioinformatics, and requires another team, statistical team, who can do the statistical genetics. Another feature of having collaboration in consortium is that it allows for larger sample sizes, which is very important. I'll show you for many studies. It increases the power of our associations. And then also, these kinds of collaborations can facilitate the inclusion of understudied population and groups, which I'll show you also in a minute. So I'm going to give you two examples today, one from early onset glaucoma genes, so the kinds of genes that affect children, and the second from primary open-angle glaucoma, which affects adults. So first, the children. In the US population, I know this would be different here in India or in other populations, but in the US population, the genes that are known to cause early onset glaucoma only account for disease in about 20% of people. So that's wonderful for those 20% of people. We can offer excellent genetic testing, but for many people that come to us for genetic testing, we can't give them useful information. One of the reasons, or one of the concerns that we thought that was limiting these results is that many of these studies have been carried out in US and white Caucasian populations. So we thought it would be interesting and useful to expand this kind of genetic discovery to ethnically diverse populations. So one population that we had the opportunity to study a few years ago was from the Philippines. And this is a picture of Ryan Calantes, who maybe some of you know, who came to my lab and worked for 5 years on a collection of families that he brought from the Philippines. These are families all with juvenile open-angle glaucoma, and we did a sort of a traditional approach to gene discovery where we did whole exome sequencing, looked for mutation in known genes, found some families with myocilin, and then looked for new genes. And interestingly, we found that 3 of these families had mutations in a gene called EFEMP1. And these are the 3 families. You can see 2 of them are really quite large and 1 smaller family. So these variants that we found in EFEMP1 were all highly evolutionarily conserved and quite likely to be the cause of disease in these families. Now, you may have heard about this gene before. This is also called fibulin 3, and this is a gene that causes Duane honeycomb retinal dystrophy. A particular missense allele, one missense mutation, causes Duane's. There was also a family a number of years ago with a rare EFEMP1 mutation, a POAG family, and common variants in this gene have been associated with IOP and POAG and GWAS, but this was the first report of EFEMP1 mutations in early onset glaucoma. And we think that we see this because this is something that exists in the Filipino population and not in the US population. We showed that these mutations cause intracellular protein retention, a functional study, and we have a genotype-phenotype correlation that we think explains the difference between why you would get glaucoma or Duane's if you have mutations in this gene. But I'm showing this here really to emphasize the point that we would not have found this result if we had not studied the families from the Philippines. Now, the second example I want to give you is primary open-angle glaucoma. And to find genes for this, we first formed the Neighborhood Consortium, which included sites, again, all over the United States, and included ophthalmologists, geneticists, bioinformaticists. This was a very successful collaboration. We identified 3 new loci for POAG, and we're fortunate to publish this in Nature Genetics. But at this point, it's still explained just a small percentage of heritability for POAG. So we expanded this to form the International Glaucoma Genetics Consortium. And this map of the world shows the names of the cohorts from all over the world that have been included in this collaboration. Putting all of this together, we ended up with many, many cases and controls, 37,000 cases, which gave us really good statistical power to find genetic variants, and also improved our diversity in this study. While we still have many European Caucasians, we were able to include a number of Asians and a number of Africans in this work. Because of this, we ended up not finding just 3 primary open-angle glaucoma loci, but 127, and expanding across the genome, so greatly expanded the number of loci that we found and the significance of the work. Also, this very large sample size allowed us to do a number of other kinds of analyses. We could do pathway studies to see like which pathways are important in primary open-angle glaucoma, things like laminar fluid shear stress and lipid transport and so on. Interestingly, we were also able to look at the difference in association effects between different ethnic groups, so comparing Caucasians to Africans and comparing Caucasians to Asians, which that also gave us some really useful information that we would not have been able to do if we hadn't included these other groups in our study. The International Glaucoma Genetics Consortium has been incredibly successful, both before and after we completed that GWAS. We have more than 30 publications from the consortium. It's supported many collaborations. Just kind of the nuts and bolts of it, we have monthly meetings and an annual in-person meeting at ARVO. But forming this consortium really allowed us to make a lot of progress in primary open-angle glaucoma genetics. So to summarize, international collaborations have had really important impact on glaucoma genetics research. It allows for larger and ethnically diverse studies. The example that I gave for that is this International Glaucoma Genetics Consortium Trans-Ancestry Meta-Analysis. And also these kind of collaborations allow us to look at unique populations, for example, the Filipino juvenile open-angle glaucoma and our discovery of EFEMP1 as a causative gene. So I just want to acknowledge some people. This is, first of all, a lot of people from the Philippines who worked hard on that study for EFEMP1, our neighborhood consortium, and then all the people who are in the International Glaucoma Genetics Consortium. Thank you very much.
Thank you, Jenny. Anybody wants? Yes, Kumar.
Thank you. Good talk, Vinny. So the question is about the fibrillin 3 gene variant in glaucoma. So have you followed them up to see if they develop any retinal degeneration?
Yeah, that's a really excellent question. And I kind of went over that fast because I didn't want to go to, oh, it looks like it's not here anymore. But it turns out that the variant that causes adult onset POAG, so there's three variants. Or three classes of variants. There's variants that cause severe early onset glaucoma, like juvenile. There's a single variant that causes adult onset disease, so not severe glaucoma, but adult onset. And then there's the variant that causes Doyne's honeycomb dystrophy. It turns out that the POAG variant, those people have both glaucoma and retinal dystrophy. But at the ends of the extreme, the severe glaucoma people do not have retinal degeneration, and the people with Doyne's honeycomb dystrophy do not have glaucoma. And we think it has to do with how much of the protein is sequestered within the cell. And so the POAG one is intermediate.